Hi, gene regulation students, and welcome to your next lecture. In this lecture, we'll continue with part 1 on the structure of DNA and epigenetics. In this lecture, you will learn to explain how DNA methylation affects gene expression, discuss how DNA methylation can be investigated using the techniques methylation-sensitive PCR and bisulfite sequencing. We will also have an overview of genomic imprinting and X inactivation as examples of how structure affects gene expression. And then we will discuss some of the mechanisms by which noncoding RNAs, specifically long noncoding RNAs, contribute to transcriptional regulation. In the previous 2 lectures, we looked at chromatin structure. We also looked at how histone modifications can alter chromatin structure. And then we had some examples of chromatin remodeling complexes, such as the Swiesniff complex and the HMGNs. Now let's discuss DNA methylation. DNA methylation is another chromatin modification, and this modification involves the addition of a methyl residue to position 5 on cytosines to form 5-methyl cytosine. As you can see here, this is the benzene ring of cytosine, and a methyl group has been attached to the carbon at position 5 in the benzene ring to form 5-methyl cytosine. 2 to 7% of mammalian DNA is modified with methylcytosine, and 90% of these methylated cytosines have an adjacent 3' guanine, as depicted in this part of the diagram. These are referred to as CPGs, or can also be called CG dinucleotides. The P in CPG represents the phosphate group in the sugar phosphate backbone. Studies have used methylation-sensitive restriction enzymes in order to investigate how DNA methylation impacts on gene expression. A methylation-sensitive restriction enzyme is able to cleave DNA that is not methylated, but is unable to cleave DNA that has a methyl residue. In this way, it is possible to investigate which residues are methylated and correspond this to the expression of genes. From these studies, we have found that increased DNA methylation is associated with inactive genes, and decreased DNA methylation is associated with active or potentially active genes. Gene promoters have also been shown to have areas rich in CG dinucleotides, or CPGs. DNA methylation can also be investigated using bisulfite treatment. Bisulfite is a compound that is able to convert unmethylated cytosine residues to uracil. However, bisulfite has no effect on methylated cytosine residues. After treatment with bisulfite, DNA can be exposed to hybridization probes. These probes can be designed to target specifically methylated or unmethylated DNA. For example, if the DNA was originally methylated, the DNA would have a cytosine residue at the same position after bisulfite treatment. However, if the cytosine residue was not methylated, bisulfite treatment would convert that cytosine to a uracil. As you can see, there's a change in the structure of DNA after bisulfite treatment. Therefore, a hybridization probe that uses G in the position to target a C will bind to methylated DNA sequences. However, if the sequence was unmethylated, the hybridization probe containing an A in the position that was matching to the original cytosine will now bind to that DNA that was treated with bisulfite. So guanine will form a complementary base pair with cytosine, and adenine can bind to uracil. It's important to take note that bisulfite treatment can result in a high level of DNA damage, and so this treatment can be used in combination with other techniques. For example, methylation-sensitive PCR involves pre-treatment with bisulfite, and then PCR primers or probes are designed to target specifically residues that have either been methylated or unmethylated. If the primer contained a G in the position of a methylated cytosine residue, it will not hybridize to a region that has a U in the position of the cytosine. However, if the probe was designed to contain an A in the corresponding region, it will bind to unmethylated DNA sequences, but not to methylated DNA sequences. And this will result in amplification or no amplification, depending on the scenario. Bisulfite sequencing uses a similar principle, but rather than looking for a PCR product, this technique is used to directly sequence the resulting DNA. More information on methylation-sensitive PCR and bisulfite sequencing has been provided after this video. DNA methylation is catalyzed by a group of enzymes called DNA methyltransferases. This can be shortened to DNMT. DNA methylation patterns have been shown to be propagated through cell divisions, which means that after DNA replication and cell division, those methylation patterns are maintained. DNMT3A and DNMT3B catalyze de novo DNA methylation. By de novo, we mean new. So this means that DNA that was not originally methylated can now be methylated by these particular enzymes. While DNMT1 catalyzes methylation of hemimethylated cytosines. By hemimethylated cytosines, what we refer to is after DNA replication and cell division, only one of those cytosine residues will be methylated on the DNA double helix. And so therefore, DNMT1 can catalyze the methylation of the cytosine residue on the opposite DNA strand. DNMT1 is involved in the maintenance of methyl marks. Therefore, methylation can be maintained after cell division due to the presence of this enzyme DNMT1. DNA methylation is also a reversible process, and there are a variety of enzymes that can catalyze the demethylation of DNA. DNA methylation has been shown to be linked to large-scale chromatin modifications. And that's because specific proteins can be recruited and bind to methylated DNA specifically. An example, MECP2, is a protein that specifically binds to methylated CpGs. When MECP2 binds to a methylated cytosine residue, or CpG, it recruits a histone deacetylase. As we've learned in previous lectures, histone deacetylases are involved in the removal of acetyl groups on lysine residues within histone proteins. Since histone acetylation is involved in loosening of DNA, deacetylation of histones is involved in tight packaging of DNA and therefore gene repression. Therefore, in this way, DNA methylation can be linked with large-scale chromatin modifications due to the recruitment of histone-modifying enzymes, and therefore DNA methylation is tightly linked to chromatin modification. Now I'd like to discuss another concept called genomic imprinting. There are a few number of genes that are involved in monoallelic gene expression. When we talk about monoallelic gene expression, we refer to a situation where only one of the two alleles present in the genome is transcribed. Now, as we know, we inherit two copies of chromosomes from each parent, a maternal and a paternal set of chromosomes. And in most genes, the expression from both alleles is required in order for adequate gene function and protein production. However, in a selected set of genes, only one allele is expressed, and this is called monoallelic gene expression. Since only one allele is expressed, the other allele generally has to be silenced, and this pattern of silencing is referred to as DNA imprinting. DNA imprinting refers to a methylation pattern or mark that is created early on in embryo development. In pre-implantation embryos, majority of the cytosine residues are unmethylated. However, DNA methylation patterns are created early on during embryogenesis, and this is regulated by the presence of histone modifications. Therefore, what this tells us is that histone modifications can regulate DNA methylation, and in the previous slide we saw how DNA methylation can regulate histone modifications. Therefore, the process of DNA methylation and histone modifications are tightly linked to each other. In the case of genomic imprinting, in pre-implantation embryos that do not have methylated cytosine residues, some regions have been shown to contain methylated lysine on histone 3, or H3K4. This methylation pattern of lysine residues on histone 3 can regulate which regions become methylated and which regions do not become methylated. As I've mentioned in a previous slide, DNA methyltransferase 3 is involved in de novo methylation patterns. In pre-implantation embryos, DNMT3 works in combination with another enzyme called DNMT3L. DNMT3L is involved in regulating which cytosine residues get methylated by DNMT3 by binding to lysine 4 on histone 3. When DNMT3L binds to this lysine residue, it recruits DNMT3, which is involved in DNA methylation. However, if lysine 4 on histone 3 is methylated, DNMT3L can no longer bind to that lysine residue, and therefore, regions of the genome that contain methylated lysine 4 on histone 3 do not become methylated on their cytosine residues in the DNA. This tells us that the pattern of H3K4 methylation that is present in pre-implantation embryos are involved in regulating which regions of DNA get methylated and which regions of DNA that do not get methylated. In females, monoallelic gene expression is most commonly observed in the case of the X chromosome. Due to the fact that females have two X chromosomes and males have an X and a Y chromosome, there are extra genes present in females due to the presence of two X chromosomes. Therefore, one of those X chromosomes have to be switched off in order to allow for monoallelic gene expression in order to correspond with males. This dosage compensation is achieved through X inactivation. When we talk about dosage compensation, we mean that the expression of the genes on two alleles is too much, and therefore one of those alleles have to be switched off. Therefore, in females, one X chromosome is inactivated, and the inactivated X chromosome takes the form of a bar body. The bar body is an X chromosome that has been highly heterochromatized or repressed in order to prevent the expression of genes. Studies have shown that inactivated X chromosomes are hypermethylated, which means that the entire X chromosome contains methylated residues. One example is the PKG gene, which is located on the X chromosome. Methylation analysis of this gene has shown that 60 out of the 61 CPGs are methylated on the inactive X chromosome. However, all these residues are unmethylated on the active X chromosome for the same gene. 5-azacitidine is a drug that can be used to remove methyl marks on cytosine residues. Treatment of cell with 5-azacitidine resulted in reactivation of the genes on the inactive X chromosome, and therefore we can see that methylation of the X chromosome is tightly involved with repressing genes on this chromosome. In addition to DNA methylation, inactive X chromosomes have also been shown to have other chromatin modifications, such as reduced histone acetylation and reduced methylation. All these modifications are associated with a tightly packed chromatin structure, resulting in inactivation of the X chromosome. Now that we've discussed DNA methylation and genomic imprinting, I'd like to move on to how non-coding RNAs play a role in regulating gene expression. Short non-coding RNAs are RNAs that are less than 31 nucleotides, and these have been shown to repress gene expression. The mechanisms by which short non-coding RNAs are involved in repressing gene expression will be discussed in further lectures. In the case of this lecture, I would like to discuss long non-coding RNAs, or LncRNAs. LncRNAs are transcripts that are longer than 200 nucleotides. These non-coding RNAs differ from mRNA due to the fact that they lack an open reading frame, which means that these long non-coding RNAs are transcribed, however, they are not translated into proteins due to the lack of an open reading frame. Long non-coding RNAs are involved in regulating the expression of protein coding genes. They are also named from the position that they are located in proximity to the protein coding genes that they regulate. For example, antisense long non-coding RNAs are located in regions that partially overlap protein coding genes. So this diagram here is showing the position that long non-coding RNAs are transcribed from in the genome in relation to the protein coding genes that they regulate. The antisense long non-coding RNA is transcribed on the opposite strand of the protein coding gene in the opposite direction, and some of this long non-coding RNA's regions that are transcribed overlap with the regions that are transcribed into the protein coding gene. Intronic long non-coding RNAs are transcribed from regions that are located in the introns of protein coding genes. Bidirectional long non-coding RNAs are able to use the same promoter as a protein coding gene. This tells us that the long non-coding RNA is transcribed at the same time as the protein coding gene, but in the opposite direction. So say, for example, we have a transcription factor that binds to a promoter region for the protein coding gene. The long non-coding RNA is transcribed from the opposite strand in the opposite direction. So when this transcription factor binds to the promoter, it is able to recruit two different DNA polymerases. One DNA polymerase, usually RNA pol 2, is able to transcribe the protein coding gene, and another polymerase transcribes the long non-coding RNA gene in the opposite direction. Bidirectional long non-coding RNAs have been shown to be involved in the repression of these protein coding genes. The last category of long non-coding RNAs that exist are intergenic long non-coding RNAs. Intergenic long non-coding RNAs are discretely transcribed away from the protein coding genes that they regulate. So long non-coding RNAs are just beginning to be discovered and described, and these words or names are used to describe the positions from which they are transcribed. The position from which long non-coding RNAs are transcribed is important because this gives us a good indication as to what their functions may be. In this diagram, we'll discuss some ways in which long non-coding RNAs are able to regulate gene expression after they are transcribed. So in the previous diagram, we saw the positions from which long non-coding RNAs were transcribed. Now in this diagram, we'll discuss how long non-coding RNAs can regulate gene expression after they've been transcribed. Long non-coding RNAs have been shown to form RNA-protein complexes. Now remember, these long non-coding RNAs are single-stranded molecules, and they are able to fold back on themselves and form specific structures that can bind to proteins and DNA sequences as well. Long non-coding RNAs have been shown to regulate gene expression by associating with chromatin modifying enzymes. They can associate with these enzymes and deliver them to specific parts of the genome. This diagram shows 4 ways in which long non-coding RNAs act within the nucleus in order to regulate gene expression. Long non-coding RNAs can act as decoys. These RNAs are able to bind to transcription factors. So we will discuss how transcription is regulated in the next section. However, for the purpose of this slide, transcription factors must bind to promoters in order to initiate gene expression. So when the transcription factor binds to a gene promoter, it is able to recruit an RNA polymerase, and this results in gene expression or transcription. When a long non-coding RNA binds to a transcription factor, it can sequester that transcription factor and prevent that transcription factor from binding to its DNA target. And therefore, a long non-coding RNA can act as a decoy. So the transcription factor which binds to that long non-coding RNA cannot bind to the DNA sequence of interest and therefore will inhibit gene expression. Long non-coding RNAs can also act as an adapter. An adapter is a molecule that can bring together 2 or more proteins in a complex. So proteins can bind to these RNAs and specific parts of the long non-coding RNA. And when the long non-coding RNA binds, say, 2 or more proteins, these proteins now come into close proximity with each other. When they are in close proximity to each other, they are able to form a complex and can now exert their function in combination. In this way, long non-coding RNAs act as adapters to bring together different proteins in a multi-protein complex. Since long non-coding RNAs can bind to both protein and DNA, they can act as guides. In this way, long non-coding RNAs are able to target chromatin remodeling enzymes to specific genes for silencing. Here's an example in the form of a picture. We have a guide long non-coding RNA that is recruiting 2 chromatin remodeling enzymes or proteins to a specific location in the genome by binding both to the proteins as well as to the DNA sequence. It can also act as an adapter in recruiting other proteins to DNA binding proteins. So in this case, we have a DNA binding protein that can also bind to a long non-coding RNA. And this long non-coding RNA can recruit other proteins to the DNA binding protein by acting as a guide as well as an adapter. Long non-coding RNAs can also act as enhancers. Now, as I've mentioned earlier, in order for a gene to be expressed, it requires transcription factor binding. Long non-coding RNAs can recruit proteins such as transcription factors and transcriptional enhancers to the upstream regulatory regions of a gene in order to enhance the expression of specific genes. And so long non-coding RNAs are a new class of molecules that are now being characterized and their functions are being discovered. And these are some of the ways that long non-coding RNAs also serve to regulate gene expression. A great example of how a long non-coding RNA has been involved in regulating gene expression is the discovery of EXIST. The EXIST long non-coding RNA has been shown to be involved in X chromosome inactivation. So before I explain what LncRNA EXIST does, I'd like to explain about another group of chromatin remodeling enzymes called the polycomb repressor group. Polycomb repressor group proteins, or PRC proteins, are involved in forming complexes that play a role in gene silencing through modification of histones. There are a number of PRC complexes. PRC1 and PRC2 are the major ones. PRC complexes have been shown to silence gene expression by recruiting histone deacetylases to specific parts of the genome, and they've also been shown to play a role in DNA methylation. PRC complexes contain RNA binding proteins, and they are able to bind to a variety of RNA targets. A number of long non-coding RNAs have been shown to bind to and recruit PRC complexes to specific gene targets. So an important thing to consider is that PRC complexes tend to be promiscuous in their RNA binding properties. They don't necessarily bind to specific RNAs and have been shown to bind to multiple RNA targets. And long non-coding RNAs have been shown to bind to PRCs and recruit them to their target genes. The long non-coding RNA exist is able to bind to PRC2. Therefore, the localization of exist is important because since it can bind to and recruit PRC2 to a specific part of the genome, PRC2 will be involved in remodeling the DNA that is linked to exist. LncRNA exist is localized to gene-dense regions on the X chromosome. So it is transcribed from the X chromosome and it's located in gene-dense regions on this X chromosome. And it specifically is expressed from the inactive X chromosome. LncRNA exist has also been shown to be stably expressed during the process of X inactivation. Studies have shown that LncRNA exist is able to silence regions of the X chromosome by recruitment of PRC2. The long non-coding RNA then migrates to proximal regions on the X chromosome, which means that once one part of the X chromosome has been silent, the long non-coding RNA then moves along the X chromosome and recruits that PRC complex to continue silencing the X chromosome until the entire X chromosome has been repressed by the action of the PRC2 complex, which is involved again in histone deacetylation and DNA methylation. I hope this gives you an idea of how long non-coding RNAs can be involved in regulating gene expression through their interactions with chromatin modifying enzymes. And this brings us to the end of our lecture. In next week's lectures, we will start going through the process of transcription in much more detail, and we'll talk about gene expression.